Good evening. Happy Wednesday. Today is Wednesday. um, my name is Tekiwa Smith, founder and executive director of Science, Engineering, and Mathematics Link Incorporated. And I want to welcome you to our Math and Science Career Academy Teen Science Cafe. Um, this cafe is bittersweet because it is our final program activity for this program year, for our 2021-22 program year. We go on hiatus during the summers and we will resume programming in August. However, we won't completely go away on you. We will have things over the summer, IG stories, IG takeovers. What else will we have this summer? But mainly on our social media. And if you missed any of our previous programming, you can go through it on our YouTube channel and learn about the various STEM careers and the journeys of our STEM professionals. And we will be back in August. Also, social media, look for um, the various programs that will resume in August. Some of them you can sign up for. I guess I should have waited to the end, but I'm gonna say this now because it's on my mind. Just like for Metro Atlanta students, we are opening the application for our STEM Journal Club, our third cohort. For those of you in Atlanta, in August we will start hybrid programming, which means we're going to do a few in-person activities, mainly our Teen Science Cafe and our STEM in the City workshops that'll be open to the public. Um, Depending on how the pandemic is, we are going to have our career fair in April in person. It may be a little limited, but we are just really thankful for every one of these 2 years in virtual. We really did not know, um, when the pandemic hit, what we were going to do, if it was the end of our programming. And so we are very thankful to technology to be able to bring these sciences to you. We are thankful for the scientists from all over the country. That was the beauty of it being virtual. We were able to bring scientists outside of Atlanta and DC, the areas that we serve. And although we're going hybrid, we are still going to continue to offer virtual programming, not as frequently, but we are still going to have virtual teen science cafes. So this summer, just stay on our social media. Um, Carlin will say what we'll do at the end, but just stay engaged with our social media to resume programming. And so in August. And so I am going to turn it over to Carlin, our program coordinator, who is going to introduce our speaker.
Thank you, Tekiwa. Hi, everyone. Thank you for joining us. Tonight's speaker is Dr. Erica Harris, who is a postdoctoral researcher at Agnes Scott College. Her primary role is researching the role that animals-microbe associations play in disease and cannibalism using insects. She also advises undergraduate students on research projects and teaches integrative biology, cellular biology, and biostatistics. Her ultimate goal is to cultivate a community of critical thinkers while personally reflecting and adapting in the process. She believes in encouraging students to be good communicators of evidence-based work. So without further ado, I'll hand it over to Dr. Harris.
Hello. Thank you so much, everybody, for attending, who's attending live presently, and those who will go back and see this recording at a future time. I want to thank SimLink, primarily Tekiwa and Carlin, for even thinking of having me come and just talk to you all about things that get me really excited about science. So for today, could this be a whole lecture? Yes. But my goal is to just really get you excited about just the different possibilities that exist within ecology and really primarily in microbial ecology. And just things that I use and tools I use and questions that make me very happy, but also just really try to get you an idea of like, how does a researcher approach a problem and where do they go from there? Because critical thinking is a huge part of that. And that's not something you are just born with, right? You have to learn these things, and you want to be in a community that really promotes you to begin to think and probe you and challenge you to think. So, as my slide says, I'm an ecologist by training, and ecology, we'll talk a bit about it, but it's pretty much focusing on species interactions and how they interact with their environments. But I look at that, most people think of that in a very large way. I could be a human species going to interact with a tree or the environment. That is a form of species interactions. However, in my studies, in my research career, I look at it at a very micro scale, looking at different microbes. So we are going to look at ecological communities in and around us. Oh, my picture's pixelated. That's okay. So we live in a microbial world, and I want to start with that by saying, not only in this picture do you have a human that is here in the middle. See, my cursor does go. Okay, so you have a human in the middle, but it's not just them inhabited by microbes. So on and in their bodies, a lot of microbes, and we'll talk about those types. But it's also microbes in the grass. It's also microbes in the tree. It's also microbes. I know it's been a while. There was a group at Georgia Tech looking at microbes in the air, and they like occupy different the stratosphere and different clouds. There's microbes in the water. There's microbes in the food you eat, specifically fermented food like wine, cheese, yogurt. Microbes are used to actually produce that food. So we live in a microbial world, whether people want to accept it or not. So then there's a lot of different common types of microbes. So for today, from this point forward, when I refer to microbes, I'm going to be referring to bacteria for the purposes of this workshop. But there are different types, right? There's bacteria, there's fungi, there's protozoa, and there's viruses. And these mainly differ in, I guess, the DNA, not just DNA, viruses have RNA. So the genetic code in them, that's how they differ, but they also differ in structure and just their evolutionary history. I'll start by saying bacteria are prokaryotic, meaning they have cell walls. They don't have membrane-bound nuclei, whereas fungi and protozoa are eukaryotes, meaning they have membrane-bound different organelles, and they are just seem to be a little bit more complex. And viruses, on the other hand, they don't even, unlike bacteria, protozoa, and fungi, who typically use DNA as their genetic structure, viruses can be DNA-based, RNA-based, and they only can live solely by attaching to a host, as we know with COVID and the flu, all these things. They can only live by attaching to a host and really hijacking that host cell machinery to be able to complete its life cycle. So viruses are vicious, but other things can be vicious too. So we live in a microbial world. So one thing that we hear a lot about is this human microbiome. So in that picture I showed you earlier, not only is it microbes on us, in us, they also are around us. But most people, I feel like most humans, we are in some ways, we're a bit selfish, and we like to talk about ourselves. And I don't think it's a problem with that. I just think whenever you're learning a concept, you always kind of want to start with what interests you and how are things relevant to you. Even when you're trying to pitch an idea to somebody else, you want to tell it how it's relevant to them. So whenever I start by talking about different microbes, even though I study, as Carlin mentioned, microbes and how they interact with different insects and how those insects use those microbes to adapt to new environments, fight off parasites, sometimes eat each other. In the case of ladybugs, eat themselves. And in the case of flower beetles that I use, they eat each other. Microbes can be used for a lot of different things. So we're going to talk about some of those roles, some of those factors that drive the microbes that are on you. So the human microbiome, I think I was a junior in college when I think about when I was first introduced to this idea. The National Institute of Health was undertaken after they sequenced the human genome. They said, hey, we know it's microbes that exist on and inside of humans, and we want to begin to sequence those things. So whenever somebody refers to a microbiome, it's not just the microbes on the previous slide. And they usually are talking about bacteria, but it's not just the bacteria themselves, it's the bacteria and the community of all the different types of bacteria that exist in that, I guess, defined area, which can be a gut, which can be your skin. And then it's also all the genes associated with all of those microbes. So it's all the bacteria present, a microbiome, or all the microbes present, and all of their collective genomes. So we're talking about a lot of data-ish, because microbes don't have as much, as long of a genome as humans. So for the human microbiome, there are a lot of different, I guess, as you can look to your left column or a right column, these are all like the different places in which microbes can occupy and live in and persist. And then there are also a lot of different factors that drive what microbes we have where, right? So we have genetics, environment, diet, lifestyle, hormones, and industry. And this is not my figure. I'm not really sure what industry means. But I'm going to say, to me, that sounds like lifestyle. I'm not sure what industry means. But with that being said, things that I use in my research, and I would not necessarily really get into today about my research, but we'll talk about it in a general terms of humans and how it relates to their environment. People really care a lot about how diet affects the gut microbial community, because it seems to be, when researchers look, diet and genetics are huge drivers, and mainly diet, because when you think about that, you're ingesting, your diet is full of different substrates for chemical reactions that then can be catalysts for a lot of different things going on in your body. And based on the nutrients you feed it, whether it's full of fat or lack thereof nutrients, full of fat, full of salt, full of fiber, those things have different, those different nutrients, they have different, what do I say? I don't know. They just have different effects on your body. We'll go with effects. Okay. So I like to talk about microbes because I like to let people know microbes are super important. And not only are they important to science, they are becoming increasingly popular in pop culture. So like I said, when I was a junior in college, and that was now a little over 10 years ago, we were just still sequencing the human microbiome. But now it's really open in people talking about that. So when I say that, I mean, so up in this top left corner, I've had the privilege of going to this museum in Amsterdam, Netherlands. It's the first and the largest museum dedicated just to the history of microbes. So no, it doesn't have to be like, oh, a cow has these microbes in its gut, or a human has this. As you see with the students or children looking at the wall, it's straight microbes. And it's a great interactive museum. They have like certain technology and interaction you use with a Nintendo Switch. It's really fun. If you're ever in Amsterdam, I highly suggest it. Probiotics. I know it's virtual, but if there were a virtual show of hands, if you can just recollect, have you ever heard the word probiotics? I think most people have, right? And probiotics are actually, we have these things called good microbes and bad microbes, right? That's how people try to classify them. So the idea with the human microbiome is that not all microbes are bad, and that microbes can actually be used to help fight off infection, and fight off, and I apologize, I live near a fire station and I live near a train, so that may be a thing. And it's gone. Okay, so with that being said, probiotics are actually pretty common. When I think about probiotics, you see them on the shelf of your grocery store. You can buy them off Amazon Prime. But what I think is interesting about probiotics is that scientists to this day, especially when it comes to humans, do not really tell you the mechanism in which probiotics are beneficial. And of all the bacteria out there are only a few in the genera of Lactobacillus and Bifidobacterium that seem to even be beneficial to different health, different aspects of our health. And that's another thing that's really great in the research is when it comes to humans and probiotics, people say, well, they're good for your health. And it's like, they say your digestive health. And I guess I would like to think of probiotics maybe helping us maintain like a healthy homeostasis, balance in our bodies. But all of that's not really well known. And you would think it would be. So there's more research to happen there. But most research with probiotics has been happening in aquaculture with fish, with shrimp, with tilapia, and in a lot of cattle. And people really only care about cattle when it comes to sheep and cows because of food production. So one of my favorites is that there's this thing called Open Biome in Boston. It's a company based in Boston, but microbiomes and the benefits to microbes in helping cure certain diseases, particularly Clostridium difficile, which we won't go too much into today, but because of the different, I would say, even irritable bowel disorder, there are different, inflammatory diseases that happen in your gut that microbes have helped and shown. And clinically, these things have proven so well that there are startup companies, this Open Biome, I know it's in Boston, where you would think of giving like a blood donation or, I would say a blood donation. They actually take stool samples. And I, just for fun, one day I went online and I did the screening because I'm like, man, you can get paid to give fecal matter away. And yes, you can. However, they are super strict on you being healthy. So I took the screen just because I was curious. And these are the things I do sometimes in my spare time. Come to find out, my stool was not healthy enough to be considered as a donor. So that hurt my feelings, but it's okay. It didn't really hurt my feelings, but people are getting paid to donate stool. Other ways in which we see a lot in the pandemic, we know microbes can be transmitted through lack of washing hands. So then you wash your hands, but then if you wash your hands too much, then you can get rid of super beneficial microbes that exist on your skin surface. And then two books that really I highly recommend if you're just even interested or remotely interested in just the conversation of microbes and their ubiquity or their just being very common and everywhere. I highly recommend this I Contain Multitudes book by Ed Young. So much that this book told me about this museum, and I went to the museum when I was in Amsterdam. But also, if you care more about microbes in a health perspective and how the absence of microbes due to the overuse of antibiotics, which are again are microbe killers. Mainly, they're produced by bacteria. They kill bacteria. Highly recommend this book by Marty Blazier. And they both just go through a lot of case scenarios. So I like books that they'll refer to a lot of research papers, but they break it down and it's not super scientific jargon. Culturelle, another different probiotics. You can go to any store and these things are on sale. You usually have to take them daily. And I do not have the video clip for this up because every time I try to show it, the link acts weird. But I highly recommend if you're into South Park, there's an entire episode. And it's pretty scientifically accurate. It's also South Park, but it's pretty scientifically accurate on stool samples and fecal transplants. And of course, there is some comedy in there and talking about they were trying to get like the perfect. So remember, I told you I wasn't a viable donor for stool for open biome. In South Park, they were trying to figure out who would be the most healthy donor, and they concluded that that was Tom Brady. But I'm sure we can conclude of a lot of different people who we may think, but that is just fun. So microbes are in pop culture. So a huge thing that I do before we even try to start talk about how people ask questions or how scientists and researchers ask questions, and you all, I'm considering you all scientists as well. You may go in and you may want to know about different communities and microbes. And microbes never exist like by themselves. They're tiny. They are microscopic by the name. They never exist by themselves. They are usually within a community, and that's where my ecology background comes in. I really do enjoy it because as an ecologist, I am trained to assess communities, but it's macro communities, right? So in ecology, a community is you can go into a forest, right? When I took ecology class in college, that's how we went. You know, we go on campus, we start to measure out different quadrants and see how many trees exist in this quadrant. A quadrant can be a foot by a foot, a quadrant can be 10 feet by 10 feet, right? You were just assessing how many species or how many species, living organisms, like live within this area. So in ecology, we take a lot of those big macro principles. We do trees, we do humans. You could do, if you're doing conservation, you can say how many sea turtles are on this beach, right? But these are what I like about this growing field and an interest in microbiomes is that there are long-standing ecological principles that have been in place. And every time someone is assessing a microbial community, we're about to go into that, it may not be that fun, but you're going to do what the researchers do. But when we get into those things, when we assess microbial communities, all of those techniques are taken from ecology, the field of ecology, and it's taken from historically what has been used to assess communities, usually, like I said, for conservation reasons. All right, so how do we assess a microbial community? So there are going to be some research and some data that we're going to look at the end of this presentation, and we're going to go into a database online that I like to use as a teaching tool with different data sets, and we'll talk about it. But with those things being said, I first want to talk about what we will be looking at and just really on a simple model terms. So to characterize a microbial community, it's a few terms that people use, and I just want to introduce you all to those terms. So people talk a lot about community composition, and that typically when you think of composition, it's like, what is something made of? What is a community made of? And that's typically who's there and how many are there. So I'm going to give you two hypothetical communities of community A and community B. Right now, we have 4 key players in our community A and community B. We got an orange hexagon, a pentagon. We got a yellow circle. We got a red, purple. Oh gosh, I do not know my colors. A blue triangle and a purple diamond. But with these things, I know although simple, that's why I like models. You can always say, hey, each of these shapes can be bacteria A, bacteria B, bacteria C, bacteria D. These can be different humans. These can be different tree species. These can be different insects. These can be, we'll stop there. They can be those things. So community A and community B, if we think in terms of community composition, at a basic level, like I said, we just ask it who's there and how many is there. So I just described you, we got 4 different shapes in different colors. Even if you want to get a little more complicated and do some variations, you could have, instead of a blue triangle, you got a blue triangle and a green triangle. So that's saying, hey, those triangles are more related to each other than they are to the circle in this. Okay, so if we look at community A and community B, we would simply, community composition would be us going through, community A has this many yellow circles, going through each shape and saying how many and saying the identity of those. So if we just look on who's there and how many are there, community A and B are different. If I tallied up those marks, right? Community A, like I said, they got different number of circles, different number of triangles, different number, same number of pentagons, and same number of diamonds. But even though some of those species or shapes are the same, they still differ in ways. Another metric that ecologists use a lot is community diversity. And I laugh because we hear diversity a lot in work, just in this day and age when it comes to diversity, equity, and conclusion. And I think, I think diversity is super important. We know that, so there's an E and an I on that diversity, there's equity and inclusion. When you talk about in terms of ecology, diversity, it has two different metrics in it. So diversity typically comes from who's there, how many are there, but it's like relative. The how many is relative to the other shape. So you get the other species present. So we usually hear diversity in terms of race, in terms of religion, in terms of gender. But in ecology, similar, but we care a lot about the spread. Who's there and what's the spread? So believe it or not, we have community A and community B hypothetically. And although in these cases I gave them both the same number of shapes, B is more diverse than A because B has more different types. And also, so A, it has a large spread of yellow, and that's a term that goes into species evenness. But we're not talking about that today. But it's spread and type go into this metric of community diversity. And it's a Shannon matrix thing, and it's a lot of differential equations that goes behind this. A lot of ecologists, fun fact, are actually mathematicians, the ones that really make up the models from back in the early 1900s. A lot of them are mathematicians. So you can be in math and apply that and be in any subject you want to be. So that's the M in Simling. Okay, so another characteristic of microbial communities that scientists look at a lot and they discuss a lot as a metric in papers is this idea of microbial abundance. And it really is just how many of a bacteria is there. I know you can say, well, who cares about that? But it actually does matter. So when you're doing science and you're looking at different samples, by the time we get from sampling to processing those samples, to getting them sequenced, to getting those results back, you're going to get signatures of a lot of things. So if I didn't assess microbial abundance, I could be naive to think that hypothetical community A and community B have the same numbers because they would both have yellow circles, they would both have purple diamonds, and they would both have blue triangles. However, as we see the full picture looking at this screen, right? And we see that there are way more yellow circles in community A than community B. And by doing microbial abundance, so community A has a lower microbial abundance than B. That may be switched. JK. Community B has a lower microbial abundance than A. The text is not right, but that's that critical thinking. But I also wanted to put up, there are different technologies used to do this. So I know going through shapes, going through definitions may not seem that fun, but when you're reading a paper, when they say, hey, we're doing bacterial 16S sequencing, we're doing qPCR, those are just fancy ways of saying, hey, we assess community composition. Hey, we can assess community diversity. And qPCR is just a way of counting microbes and getting that real number of abundance instead of just saying we got some signatures. We have genetic residue of who was there. It's like, hey, these are the ones that are here and active, and these are the numbers that are there. So that's where we'll go. So the fun part, how do these community characteristics translate to research? So before I click next, I'm going to show you a figure. Do not be overwhelmed. And I just say this because figures can always overwhelm me if people don't equate me to what are we looking at? What is my x-axis showing me? What is my y-axis showing me? But I want you to keep in mind, we'll go back a little bit, before we talk about these figures and before we start to look at databases online, I really want you to just think about, hey, and I know we only have an hour. So a lot of things I'm telling y'all, it's a whole dissertation worth of stuff. Not this, but these are the basis of things. So I'm proud of you all. We're going to do this in an hour and just kind of get a glimpse and see, hey, is this something I'm remotely interested in? I want you to keep in mind, we're just looking at community composition, community diversity, and microbial abundance. So how do these community characteristics translate into research? So I'm going to show you an example, and then we're going to do another example in a database. Do not worry. I just want to show you a figure in a paper that was in Science Translational Medicine. And I know some of you all are in the SimLink Journal Club. So this may not even, you may be able to analyze this figure without me even talking to you. But I got to give you some context, right? So we're looking at the microbial and dietary succession viewed in infants and babies for the first 2 years of their life. We are not going to go into a whole lot of detail of this. I just wanted to show you, we go from shapes, the shapes that we learned 2 slides ago. This is how this translates into a paper. But it's really just talking about the shapes, and those shapes would be bacteria and different bacteria, different bacterial species or genera. We then have to visualize that data. Usually, and what we're looking at right now, we mainly have these stacked bar plots that are showing us relative abundance, and we are looking at just like the change at the bottom. It's like a correlation chart that's showing us the change in community composition over time. Like I said, it's not the focus of our talk today, but I just wanted to show you, don't be overwhelmed when you see these figures in a paper. And this is basically just like looking at babies over the first 2 years of their life. And it's broken down into, you got a lot of different panels. Usually this is just one figure, but we have 6 different panels in this figure. And it's looking at, hey, as a baby get older, and if you were just to look at the colors, you would say, wow, in panel A of this figure, the community composition is changing over their lifetime. And how do you assess that? Once again, how we talked about those shapes and those colors, right? I like to break down things really simple when I'm learning and when I'm teaching. So if I look at a paper, I say, oh, the length of these stack bars are changing. Purple is tied to proteobacteria. We see that in our key down here. It's tied to proteobacteria, which is the Enterobacteriaceae. And this is at the phylum level. So we think about taxonomy. King Philip came over to eat good soup. We are at that first King Philip. We are at the second highest level. And we usually do our analysis. In some papers, this is probably early on into microbiome community analysis. But I typically, when I publish papers, I speak at the family level and the general level. And that's for good. And that King Philip came over for good soup. So you already know, because you all are smart. You all do journal club. You're just looking at like, hey, these shapes, lengths, these bar lengths, and shape colors and what's dominant, where red is tiny and 0. Wow, red is really blooming and blossoming by month 24 or 2 years of age. Where purple was really dominant in year 0, it's barely making a scene in, I would just say from basically like month 10 on, right? And we see that decrease, right? So when scientists begin to do these things, and they usually, you come in, you have a question, right? We're doing our scientific method. We have a question. I imagine these scientists came in and they said, hey, we want to know how the bacterial, and I think in this one, it was the gut bacteria. Well, actually, yes, it was the gut bacteria babies. And they mainly did it by fecal samples. And we'll talk a bit about that later. But they assess microbial communities by taking that feces, and we talked about that with open biome earlier. So usually scientists do feces because it's a non-destructive way in sampling microbial communities. However, that is different from what I do with insects, mainly in lab. It depends on the questions I'm asking. Sometimes I collect feces depending on how big the insect is, but it's other insects. If I want to know the gut bacteria community of a small larva that's a flower beetle, unfortunately, I am going to have to sterilize. I'm going to have to destruct the flower beetle. I'm going to have to open it, dissect it, and then I'm going to have to assess, extract DNA and do DNA amplifications and sequencing on a small gut sample. However, we think about science broadly. With humans and with most animals with a vertebrate, you cannot do destructive sampling. It's unethical. In a lot of ways, it would be murderous. So we look at fecal samples. And with that being said, they looked at fecal samples. And they basically, what I like about panel, it's really this figure in panel B, C, D, E, they're starting to do comparisons, right? They're no longer looking at that 24 months, which is 2 years of life. They're just looking at that first 6 months. And usually that's just experimental design and the way money constrains you. So you have to really say, hey, what are some questions that interest me based on what's feasible with my budget, my time, and my manpower? So given that, I'm just walking you through these things. They said, hey, we're going to look at babies that were breastfed by mom. That's skin-on-skin contact, right? And babies formula fed. And then we're going to compare that to babies that were born vaginally to babies that were delivered through a cesarean section. And I know just kind of everyday conversation, we may not think about it, but when it comes to microbial transmission, this is huge, right? It's huge on how you were birthed. It's huge on how you're fed. So we can imagine that, hey, mom we're doing skin-on-skin contact straight from her body, she may be giving over some microbes that are helpful to that baby, right? There's also a lot of talk about mom also giving away different nutrients that breast milk, as well as just antibiotics. So breast milk is packed with nutrients, and apparently microbes as well. So formula fed, that's very different. If hey, for some reason I'm traveling, for some reason I can't produce milk, or I'm having some trouble and not producing sufficient amount of milk, baby's not latching on, I go get Similac. But as we see by just looking at B and D, and we're looking just at from a horizontal perspective, that matters. Like it matters if you are breastfed versus formula fed. And going back to our figures earlier, that's really just a shape and color thing. I know it sounds basic, but that's how we do figures in science. But then, hey, if I'm coming out my mom's vaginal, so we're gonna look at our columns and see like how does vaginal birth versus cesarean birth affect microbial, the gut microbial community, which is assessed by us looking at fecal matter of a baby for those first 6 years of their life. And we can imagine, hey, if a baby's coming out of that vaginal canal, once again, it's getting a lot of skin-on-skin contact from mom. If it's a cesarean section, it completely bypasses that vaginal canal and it's coming out from her abdomen, right? If I said that right, I'm not a medical doctor, forgive me. So what you can say, once again, you're looking at shapes and colors. So I'll leave that there. I think I zoomed in on figure 8 that I talked about earlier. And I zoomed in on this figure. So what I want to get at is we live in this microbial world because we're about to transition and we're going to look at some databases online. We live in a microbial world and we focused on the microbiome and its way to human health. And we're kind of going to continue to do that a little bit in our exercise, but we're going to kind of expand this idea. We're going to look at how are microbes related to humans, not just the individual, but the humans or the other individuals, and not just the individual, but the people and humans and its family and beings in his family, including dogs. And we just know these microbes exist on all surfaces and organisms. Okay, so bear with me. We're gonna transition. And I'm gonna share my screen. And I'm sharing my screen to just show you all, like I said, we got an hour. So there's only so much we can do, but I think we can make the most of this time. So there are many different tools. It's so funny. You actually see my whole bar up here. But with my bar on my computer, I have nothing to hide. You see I already, as you can see my cursor up here binging around, we're looking at this microbiome DB. So I think the DB just stands for database. And there are a lot of different, the way research funding works, a lot of different resources that people actually, it is their full-time job to build these databases and these research bases. For you to come and you access and teachers and nonprofits like SimLink and to really integrate and incorporate them into their curriculum. So with that being said, we have microbiome database. We're going to go to this. I'm going to just show you since it's up on my computer. We have this thing, if my internet wants to work, we have this thing called vectorbase. If vectorbase wants to be fun. So once again, like I say, all these are educational tools created by scientists, for scientists, and just really to teach you. And this one goes a step further past microbiome, so I'm not going to get too far off. But this one includes genomic data and transcriptomic data. So genes, microbes, insect hosts, and the actual expression of those genes, transcriptomics. We'll get back to what I said. And then you have all these different databases. We're doing a buffet. So this is the University of California Santa Cruz. I really like their genome browser. I use it a lot in different classes. Let me just show you real quick if I can find it, if I can stay focused. So in the genome browser, I like it because, as you see, we're going to get back to our microbiomes, but I'm also an ecologist, but I'm training as in ecology and evolutionary biology. So from an evolutionary perspective on this left side of the screen, we even see an evolutionary tree. So we started with humans and chimps. Humans and their relatedness to other non-primate humans, such as chimps, bonobos, gorillas, orangutans, and gibbons. But then if we scroll down, because I guess the further, usually just a rule of thumb, whenever you're using evolutionary biology trees, the further it is away from a species, the further a species B is from A, the less related they are. But I like this resource because it covers a range of animals. We go from platypus, we go from zebrafish, we go all the way down to insects, microbes, and it's this virus, fungi, what do they call flatworms and fruit flies. But this is not our focus, but I just wanted to show you there are many different databases that you can use in class and questions and tools you can use to answer things. All data does not have to be generated by you. So one thing, and kind of in the vein of our babies that we talked about earlier, and those babies that were born via vaginal birth or vaginal cesarean, and or the diet they were fed on. So we see already that birth mode and diet can be drivers of what changes or what dictates what microbes are present and the abundance that they are present in. So you see how we went from shapes to colors to sounding scientific. This is how we do it. Okay, so we're gonna click on this family microbiome. This is a cool data set, I think, if you're just going to get introduced to microbiomes. And once again, we are humans. We care about how things relate to us, inside of us, our family, and the world around us. I like this one because it's really looking at a family, or they call it cohabiting individuals. So you don't necessarily have to be genetically related. You just have to live together. And in living together, we also have our pets that live with us as well. So they looked at 159 humans. So this database is really nice. They tell you the main publication that this paper, let me see if I can click on it. This study was about, this data was about. Okay, boom. And so you click on it, you already see your primary literature. So for those of you in Journal Club, you're like, hey, I can I can tell what this is and I can recognize it. And there are figures very similar to what we were just looking at in here, but we're going to look at the data. So it tells you a quick little synopsis about the data. And then this is usually termed as metadata. So they call it description on here. But whenever you begin to dive into data, and before you dive into data, you want to check the metadata. And as you begin to create your own data about stuff, metadata is important. So what is metadata? Metadata is the data about the data. You may say, well, so we got data to go to, but you need a summary if somebody's coming in like us to say, what am I even looking at? So what are you looking at here? So I already read these things, but it's we looked at 159 different humans and 36 different dogs across 60 different families. And in this paper, family is mainly referred to as just cohabiting individuals. So your college roommate could be your family as far as this paper. It's just living in clustering of humans, right? Your family, your roommate, your significant other. I can't even think of much else, but there we go. And they looked at 60 families, 159 humans, 36 dogs. I don't know if you all have pets. You can also think of cats as well. Not in here. And they kind of grouped these families based on, did they live in the same house? Were they couples? And they then took samples, and we talked about those samples earlier. They collected fecal, oral, and skin. So when you think about that, how do you do fecal? Just like the open biome in Boston that turned me down in my assessment. And the babies that we just looked at, you literally just collect poo. And even if you ever take your dog to the vet, I come in with a bag of poo. That was recently. I thought he had intestinal parasites. Poop was looking weird. Poop is actually a big health indicator of a lot of things, in addition to telling you about microbial communities in you. So they sample poop, they sample oral, so a cheek swab, and they sample skin. That was a skin swab. And we're going to go to, so I'll tell you what they found if we want to spoil it. So this data description on this website, they'll tell you the significant findings. And for the sake of time, I'll tell you, but we're also going to look at the data. So they told us their biggest finding. So their biggest question was, we know different factors drive microbial community composition, which we talked about, and diversity, which we mentioned on the how to assess and characterize microbial communities earlier with our shapes and colors. But then there are things such as genetic relatedness. So you would expect that me and my mom, especially given a vaginal birth, given breast milk, you would imagine a mom and her baby may have similar microbial communities. So that's that genetic relatedness. And diet. So what are you putting in your body? Fibers versus fats is shown over and over again, especially in humans. That dictates a lot of the main, we'll say phylum at this point, but different taxonomic ranks of microbes that are in you and developmental stage. So we saw with our babies earlier, not only did diet matter, what phase they were in their life. And a huge point with diet and developmental stage with humans and a lot of insects, I'm biased, but a lot of animals, they're coupled together. So if I'm a human baby, I'm drinking milk. Then I started to get on table food. So that's what that 2-year span in that paper that we looked at earlier, it's kind of trying to capture that, right? That transition from milk, whether it's breastfed or formula, to table food. Then when it comes to the insects I work with. I did a lot of work on butterflies. I mean, looking at butterflies, most caterpillars, and we know them, they eat hard plants with a lot of different chemicals, a lot of nutrients, but a lot of chemicals as well to ward off herbivores or animals that eat plants. And then butterflies, they go through this pupal phase in which they're just closed up into themselves and not eating at all. And then when they emerge as butterflies, all they mainly eat I'm sorry, I just blanked. They mainly eat different like sugary substances. And that's for some butterflies get it from pollen. Very rare to eat pollen. A lot of them just suck off the I would say the nutrients, the juice in plants. Let's go there. But as you see, development and diet are tightly correlated. So in this paper, they didn't really care about genetic relatedness. They say that here. So they say in addition to relatedness, genetic relatedness, developmental stage, and diet, we want to know is there exchange in microbes with them just exchanging microbes simply because I live with you, simply because I may drink off of a cup that you say you washed and you didn't wash, simply because you can think of skin if I touch the same doorknob and you come behind me and touch that same doorknob, because none of us live in a house. I know we don't. Maybe in the pandemic, but even in the pandemic, even in shelter in place, I highly doubt we lived in a house where you touched a doorknob and you wiped it immediately. If you were in the same household with your family. They also, so we'll start there. And then dogs, you can imagine my dog, full disclosure, my dog sleeps in my bed. My dog sleeps in my bed with me. You can imagine if your dog, whether you allow it or don't allow. People say they don't allow their dog to lick them, but your dog will lick you. And they don't always take permission from you to do it. These are the things. I know people with cats and the cats tracking stuff from the litter box. So these are ways in which microbes are exchanged, touching a remote, keying on the same computer. All these things are exchanging microbes. And you would imagine if we had to make a prediction. I know this is a live broadcast and some of you all see it later, but I'm just giving y'all a chance to think. If you are scientists, you making this prediction, you have designed this study, we're going out, we've already collected our fecal samples, we collected our actually, you're supposed to make your hypothesis before you design your experiment. So we have not collected our samples. We are just curious. We said, hey, we have access to these people who are willing. We got our IRB approval. That's the Institutional Research Board that says we can go out here and we can go collect these samples from humans and they're going to let us do it. And we are going to do it in an ethical way. Would you predict, if you had to make a hypothesis, that individuals living together, due to what we just talked about in the ways of microbial exchange, would have more similar or more different microbial communities? And I'm pausing just to give you a moment to think. And for me, it's like you don't even have to be a scientist to say I would imagine they would be more similar. So what they did in this study, and what I like about this tool set, because so if you're ever interested in something like this, you can always like download the full data set up here at the top. But then if you don't wanna, you can just go click on this icon that says sample details. And it's going to tell you different things, but I'm just going to already skip for the sake of time and go to sample source here. And then I'm going to click on host information. And then host information, human or animal, because remember, we're just talking about cohabiting individuals, humans, and in this case, dogs included. But I've had students, because sometimes beginning of the semester, we take pictures of our animals, and like that's a thing. It's like our Zoom background and things, or a Zoom placeholder. And I've had students, not only with dogs and cats, students with turtles, students with pigs, students with little guinea pigs. So you can imagine those animals aren't absolved either from this exchange in microbes. Not in this study, but just in your house. So once again, this is us breaking down and we're learning different variables that this group looked at in this specific study of this paper to see like, okay, what are some drivers? So even though they said in addition to host diet, we see that they still did look at host diet. And then looking at host diet, I mean, there's all kind of things. So they did a few things. They said, okay, we look at our baby. And then we talked about that earlier. We looked at baby breast milk, baby food table. And they just said diet. So I'm going to tell you right now, you may say, wow, Dr. Harris, that's a lot of different things. We're going to go through them. But part of that sometimes, and this is I would say, not a disadvantage, but a limitation of sampling from humans, is that it's not always consistent, right? And it's not because the science is wrong or the collection is wrong. When you are doing humans, to be ethical, there's a lot of things that I'm going to say, hey, could you give me this oral swab? Could you give me your fecal sample. I know that sounds weird, but for the sake of science, you ask these things, especially when you ask people to do diet with humans, because it's not like insects, and I can't euthanize you and cut you open or collect poop from you for a very long time. So even if I'm not destructively messing up my insect samples, I still can take away samples. I can take away. I can collect fecal samples as long as I want to. With humans, there is a cap on that. So all I have to say, most humans that report diet in these microbiome studies, they're doing self-assessments. Some people forget to write things down. Some people, they may just say, hey, my diet, that's classified, whereas other people are more specific. And they say, hey, I had milk formula, but milk formula and I guess milk formula, breast milk are different. And some people, I think they just, well, what's a regular diet though? So I'm imagining this this study defined that in their introduction. And then just food type people, vegans versus vegetarians, I'm guessing versus carnivores. They looked at host life stage. And once again, so for these humans, so we started our baby paper, we went from stage was by months, right? Because we looked over a 24-month/2 year period. Whereas in this human phase, we're just saying, are you a child? Are you an adolescent? Are you an adult? Or are you a senior? So myself, I would have checked adult. You all on here for the Teen Science Cafe would have checked adolescent, just to give you an idea. When I do my insects, right now I'm working with flower beetles, and with those, I'm working with flower beetles and ladybugs. And developmental stage is like, is it an egg? Is it that we don't have eggs? We're humans, but well, actually we do. I'm sorry, but not outside of your mom. We have egg. Is it an egg? Is it a larva? Is it a pupa? There's none eaten stage, and that's for those who do complete metamorphosis. Or is it a full-blown adult? So stage matters based on species you look at. Body habitat, that's based on what you sample and what you look at. We know humans have way more body habitats than the colon, and that they're referring to, that they're relating or associating the fecal samples with the colon. Oral activity, that mouth swab, or the skin epidermis. They look at sex, but not many options. But what I cared most about, and what the purpose of this paper was really to try to look at what is the relation of self or individual to the person they were being compared to, right? So single people by themselves, and that's probably a control, right? If I'm designing a study and I care about microbial communities, switch between individuals and their cohabiting individuals, I care about, I need to have a control and just see if you live alone, what do you have? We're going to go through this slide. I see the interest of time. We're going to wrap it up. But then I want to know what is this person, their partner, their father. So basically, so it's just self, and then it's you to your partner, and then they start doing these pairwise comparisons. But I will go. I'm gonna wrap it up, and we're gonna have our conclusive Teen Science Cafe. But what we do find, or what they found in this study, is that members of the same family, and I'm just reading straight from here, members of the same family, and I'll highlight that for you, they share most of their microbiota. The individuals are different households, but we kind of guessed that just based on microbial exchange of touching common items. And there was especially a strong effect of cohabitation on skin compared to oral and fecal. That also makes sense. If we are skin, we are touching the same surface. We're touching remotes, we're touching doorknobs. And then dog ownership mattered more to, like, the skin microbiome was determined more by dogs than it was by your own children. And imagine if it's a baby. I mean, I'm holding you, but I usually got on clothes, but who knows what these animals do. I'm looking at my dog as I say that. Yes, so that is where I'll stop. I know I didn't get to show you data visualizations. Let me see. I may be able to. Quickly. Okay. So this website, we don't have to go through it all. But if you wanted to play around and you wanted to think about, because this is a recording, if you wanted to go back and think about those community matrices that I talked about earlier with shapes and colors, you can play around. So I like these publicly accessible data sets. You can do this. I don't know who's doing this in their free time, but if you were curious, and I told you, I do things out of curiosity in my free time, you could begin to see the abundance. And we talked about that, right? Community diversity, who's there and how many are there, and what's the spread of them there. And you can start to play around on this website and begin to see that related to papers that have been published. And I would say play around and then compare what you found to the papers that are published. But with that, in the interest of time, I'm going to stop. And I want to know if anybody has any questions for me. Yeah.
All right. So we did want to ask you, with you being able to share such fascinating things about this STEM area, what led you to it? What was your journey into STEM as a young person yourself?
Yeah, that's a fun question. These are things I usually talk about. But I was like, for time, I didn't want to put that in there. So, believe it or not, fun fact, I was never like that girl in high school that made straight A's. Well, I made straight A's. Okay, let me not say that. But science was like not the one that was not the subject people nominated me for an awards or whatever. I also, fun fact, I failed my first biology test in high school. But I was always interested. I'm one of those people, I let my interests guide me. So I started to do summer programs throughout, started in middle school. I started to do like the Duke Talent Identification Program. And in high school, I found out, oh, in high school, you can apply to colleges and they have these programs. And usually they ask you to pay, but for anybody on this, they also have a lot of financial aid. You just have to ask the admissions office. But my favorite one I did, and like the one my junior summer going into my senior year, was at MIT, the Massachusetts Institute of Technology. And they have two different very robust programs they do for high school students. And man, we did research. We went to this place, the Broad Institute, and they basically sequenced a third of the human genome there. So I was introduced to science research, it was a buffet. I did robotics, I did genomics, I did a lot of different things. And that made me say, hey, I want to try to pursue a career in research. And in college, I just started to do different research fellowships and got my hands wet in different wet labs.
Wow, that's awesome. And another question. So it's my understanding that there is some student science projects, events going on right now. So I was wondering, have you come across or coached or mentored any young people on STEM fair projects that are related to either microbes or just some cool biology ones that you're familiar with?
Yeah, so I think you're referring to the International Science and Engineering Fair happening right now. So I typically, with just my trainer, I typically work with college students just because that's in grad school. I was given undergraduates. There's a lot of different things you can and can't do with high school students because they're minors and they're in school. When I'm working, they're in school. So with that being said, I've actually worked with a lot of college students, and just putting them on different projects, a lot of different experiments. I've actually co-authored papers. I don't say co-authored, but yeah, co-authored papers with college students and working on these different microbial systems, and mainly with butterflies and with flower beetles.
Cool. And then last question for you was, when you were going through the diagrams, and I love how at first you were like, look, don't freak out at what you're about to see. But when students are coming into college and uh, you know looking at working with studying these type of elements, if they haven't been in a STEM journal club like we have, what advice would you give to them in being able to not be intimidated, to really learn it and feel confident in learning about it? And that even applies to just being in the lab, or any other, they say that STEM is a very challenging discipline. So just for young people who want to get in STEM, want to study the subject area like this, what advice would you give for just them being able to get over those hurdles?
Yeah, so I think that's a great question. I mean, just in all honesty, historically, STEM is a gatekeeping type field. But I think that that's part of SimLink, right? And different researchers of color out here is to make it accessible. And I don't even think researchers of color. I think people that are not of color should be making it accessible as well. I think part of it is the responsibility on educators and on different stakeholders in STEM to really to sell it to people who are budding STEM STEM professionals. I say that because it's usually taught as like, hey, you hear like those weed out classes, right? And I really, when I teach introduction classes in college, that's my chance to get you excited about something that you may have not considered or something you may have not thought you were able to do. And I think a huge part when you say like looking at graphs and whatnot, there are a lot of things I can say now in hindsight, but I didn't really feel comfortable reading papers probably until my junior year at college. But I had to kind of break it down and find me a system. And my system is that every single paper project is following a scientific method. So I always like to ask myself, what did they ask? Like, what was the question? What did they do to find it? What did they find? And why should I care? And those are the 4 questions that govern every single paper I read, every single paper I write to this day. And really just when it comes to data, like you can attack it. Some things, especially like it's in a phylogenetics, they're going to refer to genes. You just may not know what the gene is, but it doesn't mean you don't know the gene. When I read genetic papers, I automatically start giving them nicknames like gene A. I don't need to say SP500. Like that's not what I care about. I just care about gene A. So really breaking it down, doing those shapes, kind of colors, doing models that make sense to you, but they also are going to overlap a lot with the content you learn in your class.
Yes, that is excellent advice. That definitely reiterates something that I heard earlier this week too, like make it work for you, and like that's how you learn it and build that confidence. So thank you, Dr. Harris, for our last Teen Science Cafe of this program year.
Thank you for having me. I'm happy for SimLink. I hope y'all take a much needed break.
Absolutely.
Yes, it takes a lot to plan these things. And in August, we're going hybrid. And so I joke with Carla, we're having a chill summer. And not to say we won't be working busy planning for you, but, we can get to, enjoy the warm weather, think about what people we want to invite, plan our journal club, all of those things that it's better to do when you can just focus on that, right? Yes. And enjoy the summer. You want safely, because like I keep hearing there's another surge coming, right?
Coming in the fall. I was like, oh my, I need to keep my systems in place.
Yes. But whatever we can do safely to prepare, for the fall. But so I mentioned a little bit of it, Carlin. Can you give us an idea of what people can look for this summer, and maybe put our social media handles in there so people can follow us on social media?
Yeah, so this summer we will be continuing our digital learning on our social media accounts. You will be able to see us cover some STEM topics that are relevant to the summer. So, fireworks with July, with the 4th of July, as well as, the sun's rays and things like that. So we'll be breaking down those STEM areas and concepts. And also, with kids being out of school, it's the perfect time we already know you guys are on your cell phones and everything, but science communication, which Dr. Harris didn't explicitly say, but she does have, she does work in science communication. I don't know why that's coming out sounding weird, but we encourage young people to do their own science communication, with their cell phones on platforms like Instagram Reels and TikTok. And you can tag us and we will repost what you create. And finally, our IG takeovers as well, where you can hear more from people in the STEM field and, always be able to ask your questions that will help you be able to successfully pursue the STEM field. So that's all that we'll be able to look forward to.
And I want to say something about you, Dr. Harris, which one of the reasons we started TikTok STEM. I know nothing about ecology. nothing about all this stuff you're talking to, but talking about like I was a chemical engineer, but I could not turn away from you. I was like, she has so much energy. She keeps me intrigued, right? And I think that that is such an important skill to have, right? Um, so thank you for showing that. Like, you and I have met scientists all the time. We're like, I don't know what they're talking about. Um,
Right, go to sleep.
like, you literally don't know what they're talking about, and you wish that you could get that time back. But thank you for modeling, that even if you're doing something that no one has a clue, right, what it is, you can still make it interesting and break it down so we can understand a little bit. Like, maybe if I had a tad bit interest in biology, I would have been like, oh. But even my non-interested biology self was like, I can't stop listening to her. I should be checking my email. I should be doing something else. So thank you for modeling that. And don't be surprised if we email you about being a guest lecturer for our STEM Journal Club.
Okay.
So,
I love journal clubs. They're fun. So don't give me a paper to talk about.
Yeah. So whether you're live or watching on YouTube, there is a link that Carlin put in the chat for you to complete our survey. Please make time to do it. This allows us to get feedback, to know not only what topics you want to hear about, what speakers you like, who we should invite again, who should never ever invite again. But I think most of our speakers are pretty good. But it also allows us to continue to get funding to support this and to continue to host these programs for you. And so, once again, thank you so much, Dr. Harris. And Carlin put in Instagram and Facebook. And those, Carlin, just to make sure, I know we're on LinkedIn and Twitter as well, but these are the platforms, Facebook and Instagram, where they need to look for all of these things. Like LinkedIn will just be our recap, right? But all the things that you talked about, they should look on Facebook or Instagram for, correct?
Yes.
Okay, just making sure. Well, thank you again, Dr. Harris, for making time and wrapping up our 2021-22 program year. I just want to take time to thank Carlin so much for all she does as program coordinator. During the pandemic, I just was like, Carlin, you got this. Let me do some other stuff. And she has done an amazing job these past 2 years, finding people, communicating with people that we find, giving all the speakers what they need to prepare, doing the social media campaign. So I would be remiss not to end this program year thanking you so much, Carlin, for all your time and energy and making it go. So, like, all of these programs, I have been really, really working hard to do some big picture stuff. But I am so happy that we have Carlin because she can do the things that are good for you, right? The program speakers. And so far, you've been doing a really good job of picking people that are engaging. We've had a couple of hiccups, but I think this virtual is kind of hard, right? Like, not everybody can adapt. But thank you so much, Carlin. And we will see you all in the fall because we are still going to have virtual teen science cafes. We're just going to have them once a month. So look on our social media because we'll announce when we are resuming our programming. Okay. All right. And have everybody have a great and safe summer in more ways than one. All right. Good night.